Now, student, let's understand how exactly the regulation of gene expression in eukaryotes is different from the prokaryotic system. So, the control of the gene expression is more complex in the case of the eukaryotic system, as the operon is controlled by not just one promoter but more than one promoter region. And remember that in the case of the eukaryotic system, DNA is involved, because DNA negatively charged is involved in the positively charged histone proteins, which are basic in nature, and to form a nucleosome. This whole thing we have already covered in the beginning of this chapter. So that's why histone play a very significant or important role in the regulation of gene expression in the eukaryotic system. So we can call histones as the gene repressor. Now, why should we call it? Because we all have learned that the RNA polymerase enzyme, if you remember the structure of the transcriptional unit, the enzyme has to run throughout the length of the template strand. So for that, the complete strand is supposed to be made available or made loose. But we all know that histone is something around which the DNA tightly coils around. So the DNA is responsible, the histone is responsible for the masking the gene activity in non-specific manner, because histone simply switches off all the genes. Whatever gene or segment of DNA which comes under a gene is coiled around the histone will be switched off, because histone won't allow the RNA polymerase enzyme to run throughout the length, and if there is no possibility of running the whole RNA polymerase on the length of DNA, then transcription is not possible. That's why histone causes the masking of the gene activity in a non-specific manner, because all the genes, whatever gene that segment of DNA the histone comes in touch with, will cause its masking of the gene activity. And second point is the supercoiling of the DNA causes non-movement or no movement possibility of the RNA polymerase to cause transcription. So that means to cause the gene expression in the eukaryotic system, the main point is to remove the histones. So how histones are removed? So there are many scientists who have given various expression. So the removal of the histone is necessary for the protein formation, because if histones are removed, then the RNA polymerase enzyme can run throughout the length of the template strand, and this will cause the formation of the working copy or the messenger RNA, that means transcription. And if transcription successfully occurs, then only protein formation by translation is possible. So according to Paul and Steen, as well as their co-workers, in 1971 they stated that there are, NCERT also mentions this on the one of the page of NCERT has NHC mentioned there, you can check from the book. So according to these two scientists and their co-workers in 1971, they stated that there are gene specific proteins which are acidic in nature. We all know histones are basic in nature because they are rich in basic amino acids. So that's why in the eukaryotic system there are some non-histone chromosomal proteins which are acidic in nature. So they are rich in acidic amino acids, and they are gene specific. That means every gene will have a specific type of acidic proteins. Now these proteins have the affinity to join with histones, because it is very easy to understand. Histones are basic in nature, and these proteins we are talking about are acidic in nature. So because of the positive negative interaction, these proteins have great affinity for histones. So when these proteins come into picture, imagine for a second a gene has to be activated. So what will happen? Gene specific acidic proteins will go and attach with histones and remove histones from their position, so that segment of DNA, or you can say the gene, is made available for the RNA polymerase to run throughout the length and cause transcription followed by translation. And the second thing is, Britton and Davidson in 1969, they stated that that in the eukaryotes, operon contains 4 basic type of genes. So those 4 genes I have mentioned here in sequence. According to them, there is a sensor gene. Now the sensor genes are in fact the detectors, because they are sensitive to the state of the cell and they are sensitive to the environment. That means they are the one which will detect the stimulus. So these genes are stimulated by a change in the environment or change in the internal environment or external environment of the cell, and thus they are the sensors. And these genes are responsible for sensing the need for a particular protein or particular enzyme or a particular hormone to be produced. So once the sensor genes are activated, they will send a signal to the next gene called as the interrogator genes. Now interrogator genes are the genes which are associated with sensor, and these genes receive signal from the sensor gene, and they may receive signal from one sensor gene or multiple. So accordingly, interrogator genes may have many sensor genes involved or may have a single sensor gene involved, depend from case to case. And then when the interrogator genes are activated, they send the signal to the receptor genes, and the receptor genes receive signal from the interrogator gene and they signals it to the producer gene. Now what are the producer gene? Producer genes are those genes which are responsible for formation of particular required protein, enzyme, hormone. It can be anything, that means gene expression. So according to Britton and Davidson, there are these 4 genes which are involved in the regulation of gene expression in the case of the eukaryotic system. When there is a need for a production to occur, then sensor gene will be activated, which will activate the interrogator gene, which further activates the receptor gene, and finally receptor gene sends the signal to producer gene to produce the protein required.